So, in my view, George's PhD is one of the most diverse and creative and adventurous, and possibly the most beset by hardship that I have ever known. So right now, I guess you just need to settle back and enjoy the ride as he takes you through exactly what he has achieved over the last 6 years of his life.
Wow.
Over to you, George.
It's a big pressure, thanks very much.
That's right. These are the rejected permits from the first time around. Library later on.
Thanks, Rob, for the introduction. Can you hear me now in the back? Yeah, is it good now? So, yeah, I'm try to talk in these next 40 minutes about the past 6 years of my life, but focusing, of course, on the research on two large macaw species. So what are macaws exactly? And some of you that don't know these birds. The macaws belong to parrots, the Psittaciformes order of birds. And within the Psittaciformes, it's the Psittacidae family. You can find macaws only in the neotropical regions, so that's why I had to go to South America. For that very reason, the distribution is from Mexico all the way to Argentina. The special characteristic is that they have a very strong and mobile beak and zygodactyl feet, so 2 toes in the front and 2 in the back. And combining these 2 features, they can crack open really large seeds and fruits. So mainly they're frugivores, but also open like large seeds in the forest. The other characteristic is that you can recognize them. They have a very long tail and usually a big size, especially the large macaws. But there are like smaller macaws like these ones that their size is a bit smaller, but still quite large compared to other birds. And importantly, they secondary cavity nesters, which means that they don't make their own nest, but they occupy large tree hollows. So they need these large emergent canopy trees in the rainforest to nest in, and if it's not available, they can't nest. They make it larger, the nest itself, like if it's too small for them, but they don't start making a hole in the tree, and that's usually true for other parrots as well, not just for macaws. So let's see the diversity of macaws in general all the way in the neotropics. Unfortunately, we have already 6 macaws that gone extinct, mainly in the Caribbean region, but you can still see that quite a large diversity of macaws in South America. And my study species, the Scarlet macaw and the red and green macaws, these 2 macaws that are considered as least concern according to IUCN today. So the Scarlet macaw home range is really large, from Central America to all the way South America. They have 2 subspecies. The Aramacaw macaw is a South American subspecies I worked on here in Tamaulipas in southeastern Peruvian Amazon. And the other subspecies is the Aramacaw cyanoptera, which is up there in Mexico all the way to Costa Rica. The population trend, according to BirdLife, it's declining. And the population is less than 50,000 individuals around the world, but it's not, they still least concern according to the IUCN evaluation. So what are the major threats that, because why parrots and macaws are declining worldwide? The most important thing that affects them is the habitat loss. So as I mentioned, most of the parrots are secondary cavity nesters, and also like frugivores, they eat fruits and depend on the forest quite heavily. So habitat loss, meaning they're not just losing the habitat where they live in, but also the key food resources, and also the nests, which is the most important thing for the reproduction. And that's why it can act as a barrier for the gene flow and get it to losing the genetic diversity worldwide if it's like a habitat that got destructed in some parts. The other important thing that affects parrots is the pet trade. So they're very, very popular birds, as everyone knows, parrots, and have them as pets worldwide. So pet trade is quite badly affecting the natural population in the wild because of the illegal poaching of the birds still. And of course other human impacts like the overpopulation, as it affects all the other species, and urbanization, pollution, of course road effects, and hunting in some local tribes. So now I just would like to outline the structure of my PhD thesis in the first section, and there are some color codes as well, so you're going to see in blue all the sections that talk about the comparative meta-analysis we did about the psittaciformes order, the parrots, and we looked at what kind of factors affecting the extinction risk in general. Then I had the green element of the talking about the on-site conservation research, testing artificial nests in the wild in Tambopata, in Don's project. And we looked at what are the preferences of the macaws as for what kind of nest, artificial natural nest they prefer. And also we looked at the botfly parasitism in the nestlings of these birds. And from the first chapter, I start the genetic element of the PhD, starting with development new genetic markers for scarlet macaws, and validate a non-invasive so-called genetic tagging technique that I'm going to talk about extensively later for the same macaws. And the sixth chapter, I applied this genetic tagging on another macaw species, the red and green macaw, a white population in the Peruvian Amazon, and showed what kind of application we can use in the field. And in the seventh chapter, I took data from scarlet macaws, genetic data, and looked at the landscape genetics and barriers to the gene flow. So this is the outline of the thesis and also the outline of my talk today. So first, I would like to talk about the status of the parrots in general, what we found. But before that, I just quickly would like to mention or talk a little bit where they're coming from. And they're actually coming from here. They have a Gondwanan origin, according to mitochondrial DNA research. The origin dates back to the late Cretaceous, about 58 million years ago, somewhere in the Gondwana part, might be Australia or Antarctica. And then they started to radiate to the north when Antarctica started to cool down around 30 million years ago. And there were two main tribes, the Arini tribes that inhabited South America, and the Psittacini tribe that became the African parrots. And about 28 million years ago and later, several times, it happened, the colonization of the South Eastern Asians and Oceanian islands in several occasions. If you look at the diversification of parrots today, you can see that it's a very diverse system, very diverse taxonomic group. But you can look at the Arini tribe, the South American tribe, is very especially diverse today, and it's a quite old lineage. And that's probably because the uplift of the Andes mountain and providing new habitats and adaptive radiation made them very diverse in South America. And the other group that strikingly very diverse is the Lorini clade, which is the lorikeets, that live in Southeastern Asia mostly. And they are quite young group according to phylogeny, and they're probably diverse because of their feeding habits. They specialized on a niche that wasn't really occupied there before, and only eating nectars that made them quite diverse. The distribution of the parrots today, this is a map looking at the present distribution or the extant parrot species. We have about 400 parrot species living today. You see that the most diverse parrots are in South America, in Australia, but mainly all over the tropical region of the Earth. Just keep in mind that the only place they live together is 28 species, is the highest number of parrot species living in one spot all together. So that's a very rare. I think I'm going to come back to that point later. And if we look at the map where the threatened parrots are, vulnerable, endangered, or critically endangered species, you can see a different kind of distribution, but still, especially the species. Most of the threatened species are located in the Amazon basin in South America. So the status of the parrots today, if we compare this order, the Psittaciformes order, which is the parrots, and look at what's the total number of species in that order and what the number of threatened species in that order, You can see that parrots are in the second place after the Pacififormes, but it's really hard to compete with them because they have lots of species. But from the Psittaciformes, we have about 28% of the parrots that are vulnerable, endangered, or critically endangered today, and altogether 398 species. So we worked in collaboration in this first chapter with the BirdLife International, and we made we calculated the red list indices that indicates like what's the species survival, this kind of species survival index over time, and compared it to other bird groups like seabirds, game birds, pigeons. You can see that parrots are in the bottom. That means that they have the worst survival index, and just like other bird groups, they also declining over time. So this graph and all the others that I'm going to talk about now are just got accepted for publication in Biodiversity and Conservation. So IUCN evaluates all the species and put them into a category according to the various evaluation from least concern to critically endangered, and this is an ordinal scale of extinction risk. So what we looked at in this research, that we looked at first the broad trends of the threatened categories and the unthreatened categories, and compared to these species, what are the differences between threatened and unthreatened parrots. And we also looked at final details with an ordinal regression just within the threatened category to see what are the factors that push a species into a more endangered category, from less endangered to more endangered. And the explanatory variables we grouped in 4 major groups. So one of them was the geographic and distribution information about each species. We had ecology and life history information, human use, like whether they use for pets or food or for hunting. And the very novel part of this study is that we also include this socio-economical and information about the countries where the parrot distribute, and that was like the first time that research about a comparative analysis about extinction risk did this kind of analysis. Most importantly, we did a phylogenetical control of the data as well to see whether phylogeny affects any of the results, and they didn't. And actually, we find that the phylogeny of the parrots didn't affect of the threatened species, threatened status. Sorry. So you can see that they randomly distributed different kind of categories. So we're not talking about just a few taxa that are more endangered than others. It's more randomly. And thanks very much for Iliana for the great graphs. It looks really good and was featured in the presentation, in the publication as well. So what we find in the broad scale analysis, whether a species becomes threatened or unthreatened, is if they started with a small home range, so this is, here you can see the probability of being threatened, lower probability and higher probability of belonging to the threatened group. If the historical distribution size was very small, it was a very high chance that that species became evaluated as threatened than others. If you are a parrot, you don't want to be large body size because they obviously have higher chance of being threatened. You don't want to live long. Well, of course you do, but as for longevity, the long-living species like macaws, they're more threatened than the shorter-living species. The forest dependency was another significant factor that was important. Like if they were really highly dependent on forest, they were more probably being threatened. And if they lived in a very urbanized place, like a very urbanized country, the distribution, they were higher chance to be threatened. And that was one of the interesting socioeconomical indicator of the countries they actually live in. The other socioeconomic indicator was the high GDP, and that's already if a country where they lived had a high GDP, so mainly developed countries, these species were pushed into a more critically endangered category. Similarly to this one, if that species was only endemic in one a single country, like one little unit that we call a country, because of course birds like distributing in, or can fly over, but the policies of one country doesn't really do that. So if a species was only endemic in one country, that also pushed it into a more critically endangered category, and that might also do something with where these parrots are distributed. That I'm going to come back just a bit later. And about the threats, if invasive species was a large threat, that was the only significant factor of the threats. Like if invasive alien species meant a threat for some species, that meant that they're going to be more critically endangered than others. And surprisingly and contradictory what we would expect for pets, like if you think a species is used as pet, they're going to be more endangered than others, and we actually find the reverse. Like if it's used for pets, it's actually less endangered, but that's probably a secondary effect. If you think about like all the pets are quite abundant in the wild that we use, like budgies and Amazons, and so that's why the pets are it's showing like it's less concerned. That actually makes sense if you think about it, because like really critically endangered macaws are not so used as pets today. Well, not anymore. So we made all the countries into a list and called it the top 20 priority list of countries in different kind of ranks. And the first one came out that Indonesia, then Brazil, and third one is Australia, which is the first developed country in the list, a very high rank for conservation. But if you look at all the other countries, the 15 out of the first 20 belong to the neotropic regions. So that indicates us that the top priority conservation should actually focus on the neotropical parts. That's the highest distribution of the species as well, highest density of species. But fortunately, there are active conservation projects and policies in these places already. But still, it should be a future conservation a priority for these countries. So in the next segment of the talk, I'm going to talk about the nesting ecology of scarlet macaws in the wild. For this research, we used the Tambopata Research Center, or TRC, which you can see here in the middle of the Peruvian rainforest. It has a very high parrot abundance, and if you think back to that map, like the highest abundance was like 28 parrot species in one place. Here in TRC we have 20 altogether, which is really, really high diversity, and also lots of parrots abundant. It's a low elevation but very high rainfall. It's a tropical rainforest, and there's a clay lake nearby that I will talk a bit later in more details. And so as Rob mentioned at the beginning, the Tambopata Macaw project directed by Don Brightsmith afforded us a really big help and advance, because they were there from the 1990s conservation project. And in the first artificial nests that we looked at actually were designed in the 1990s, but proper data recording started with Don from 2000. So we had all this data to analyze and look at different kind of nests we used 26 natural nests in the database, 13 wooden nest boxes, and 27 PVC nests. So that's another design of nest that's made of PVC tubes that try to imitate the palm tree trunk inside. And it's more durable than the wooden one. So for the statistics of this research, we looked at 12 breeding seasons, which is quite extensive data to analyze, 145 nesting attempts. We call the nesting attempt when at least one egg was laid in that nest. And we had 5 categories of explanatory variables when we looked at information about the breeding efforts, like how many eggs, chicks were there, how many chicks hatched at the end from that nest from that year, and all kind of other information like the nest cavity characteristics or the nest site characteristics of the nest tree. And our questions were that which factor would influence the decision whether to laying an egg or not in that particular nest. and what was the hatching success and the fledging success, what kind of other measurements of the nest influenced these successes. So, and our results show that artificial nests had very similar success to the natural ones, which was quite surprising, but if you look at the natural nest success, well, the fledging was 0.6 chick per nest, where the other ones, it was quite similar to the natural ones, both the PVC and the wooden nest. The high preference was shown for successful nests. So if from a nest the previous year was at least one chick fledged from that nest, in the next year it was also occupied, so it was a very strong preference for that nest. And if there was no activity in that in a particular nest in the previous year, in the next year it was not occupied either, so it was a negative preference for those nests. And also we find that the number of initial eggs affecting the fledging success, like the more eggs they lay, the more probable that at least one chick is gonna fledge from that nest. And it wasn't so obvious before whether like the number of eggs would affect that kind of outcome. And we already published these results in the Nature Conservation in 2014. The other thing we discovered during checking these chicks in the nest is that they were highly infested by botfly parasites. So botfly is a parasitic fly that lays the larvae in the skin of the macaw chicks. And so we tried to remove these parasites using different kind of techniques that were not so successful the first time. But you can see it better now that we described a new technique just using a venom extractor device to extract that parasite, and that was the most successful of all the other techniques that we tried. We published this in the Veterinary Parasitology article, this methodology paper. And we also find that the natural nest surprisingly had lower parasite infestation than any of the natural ones, like the wooden and the PVC boxes. And our hypothesis is that it might be due to the bad thermoregulation of the artificial nest, like both wooden and PVC can heat up during the day when like the sunlight hits the nest, and because the wall is not so thick, like a natural nest, it's a very thick wall inside the tree, and it's usually dark and cool and stays like that temperature like all over the day. So that one of the idea that maybe the heat and the light attracts more parasite, more flies, and parasitic larvae for the artificial nest that can be further developed, to better enhance the thermoregulation of these nests. And the other interesting thing that we found, or actually like proof that botflies are bad for the macaws, like the more botflies a macaw chicks had, like between 0 to 63 years old, that's the lowest prediction asymptotic weight it had. So chicks with lots of botflies, they had quite low weight as well. And now I would talk about the genetic aspects of the study. For the genetics, we used a much larger study site actually than we did for the previous one. So we studied the nest and botflies in Tambopata Research Center, which is located here. And for the genetic tagging research, we sampled a huge area, like up to 1,000 kilometers per season, and went all through this river system around the Tambopata region, near to Bolivia and Brazil as well. And so that if you look at from the air, just to have an idea, the region looks something like that. It's a pristine rainforest habitat, connective habitat, protected land, and the only access to the forest is through the river. That's the Tambopata River here. So we had a motorized canoe and went through for expeditions from the research center and from Puerto Maldonado, which is the closest city in the region. But sometimes it was really hard to cross through these rapids, etc. Especially to go into intermontane valleys like this one, which is called Candamo. That's a very special part of the study, and I'm going to talk about this in the last chapter. So this is an intermontane valley bordered by the foothills of the Andes, and so it's really lots of rapids on the way to get into that habitat, and it looks something like that. You can see the Andes already around the valley. And of course you can see other species than your study species sometimes. You can see jaguars. We didn't take blood samples from them though. What we really focused on are the macaws at the clay licks. So this is a typical clay lick. Clay licks are very special features in the Amazon, especially in southeastern Peru, where we have the highest concentration, highest diversity of clay licks in South America. So other research like done studies showed that the natural food resources of the parrots in general in the Amazon basin, especially in Peru, they had a very low sodium in their natural diet. So they use the clay as a sodium intake, a sodium supplementary. That has quite a high level of sodium, and that's why probably they eat it. Another effect might be the detoxification effect of the clay itself. But for us, it was a very special place because all these birds, like sometimes you can see hundreds and hundreds of birds coming back every day to those very large clay licks. And when you go there in the afternoon, you can see lots of feather samples just left behind. Well, not feather samples, feathers and samples for us to collect and do some genetic study on them. The forest looks something like that. This very impenetrable forest. It's very hard to go just through like this and find nests, but eventually we did find some natural nest of macaws as well, so not just around the research center, but other places where no one else find or studied macaws nesting before. So when we find a nest, the first thing we had to do is climb up there, and it's really high. It's about like 40 meters high up in these emergent canopy trees. So you need to slingshot a line up to those branches and then climb up. And once you find that it's an active nest, there's a chick inside, we took the chicks like the poachers and lowered them down to the ground and pinched them on the neck and got some blood samples. And of course we put the chick back to the nest safely. But we really just needed a few drops of blood because all birds have nucleus in their red blood cells, so it contains a really high concentration of DNA to extract. And the other source of DNA was the feathers and the clay licks. And from feathers, if you look at the structure of the tip of the feather, there's a part called aparical umbilicus, which if you cut it out, you can see there is a small tiny dry blood drop inside, which contains quite high amount and quality of DNA, but if it preserved well. Sometimes the feathers found on the clay lick were quite degraded already, and of course they had degraded DNA as well, but we still use it in the lab. So after you had your samples, then magic happens, and and you get the DNA out of it. And we use this DNA for technique called genetic tagging. Genetic tagging is a technique that I'm just describing very shortly now, but just want to point out that it was mainly used before for mammals and small rodents, but never really applied as for like genetic tagging for any birds, not even like parrots or macaws. So this might be because we didn't have species-specific genetic markers on these species, and the great thing that happened in 2013 is that Dons University and the Texas A&M, the so-called Scarlet Macaw Genome Project, sequenced the whole genome of a Scarlet Macaw by a de novo genome assembly, and that provided us a huge amount of data to start designing species-specific primers, species-specific genetic markers. And we developed about 40 markers for Scarlet Macaws, we published this year. And what we did after having all the genetic markers available, we used our 9 best genetic markers, which had the most diverse markers, and applied on our study species on Scarlet macaw that they were designed for, and we also applied on red and green macaws, and they had just as diverse as for Scarlet macaw. So once you have enough genetic markers, like as I said, we used 9, you can build a genotype or a genetic tag for that individual, and it's like a forensic technique where you can use it for the unique identification of a bird. So we don't want to identify just a species, no, or just a group of species, a family group. We go down all the way to the individual level, where we can tell 2 different individual macaws just by their genetic samples, by blood or feather samples apart. So this is a very novel tool for example, population size estimate, because if you look at the clay lick closer, you can see like whatever 20 macaws on that clay lick at that point, so that's a census number, and then you come back next day and you census them again, so you have a number or an idea what's the amount of bird visiting those clay lick, but you don't actually know whether those are the same individuals coming back there or different groups, so you just see all those macaws, but you don't exactly know what's the group that visiting the clay lick. And usually if you look at from the other side, the population genetic studies, they usually just need 1 sample per individual, and they actually want as many individuals as possible in different regions to tell something about the genetic diversity or the genetic structure of the population, but they don't really interested in like the individual level. And for us, it was very important part of the research, the resampling. So once we sampled the clay lick, we didn't just like, all right, we have samples from these parts already, let's go to somewhere else. But we went back to those clay lick every like second week and resampled, and we actually wanted as many recaptures or resampling the same macaws as possible. So for example, if let's say we find 6 feathers and we extract the DNA and have the genetic tag on them, you can now see that this is one individual, this is B, C, and then you can have like, oh, this feather is from this one as well, that the same individual is coming from. So then we just call that a recapture, or like this one. Another important thing that you can do with genetics is do molecular sexing and get some kind of demography information about the parrot. So part of having a genetic tag, which individual it does, we also know like what's the sex of that bird. Like we don't even, and it's really amazing because we don't even see the birds that left the feathers behind. We just collect them, and we have all these kind of information that we can use for extensive statistical analysis later. And of course, you can all reply all the genetic population questions as well, like we ask about the sex bias dispersal, relatedness, inbreeding or gene flow in the region, that we also did. But the novel thing about the study was the genetic tagging and recovering individual genotypes. So the results of the validation of this method was we find that feather sample, it's very important what kind of feathers you collect. For example, if a feather had a good quality at the extraction, it had much better DNA for use in the lab. And another significant factor influencing the quality of the DNA was the clay, whether the feather was contaminated with clay or not. And that's like a guideline for future recollection. If a feather is like really degraded and there's lots of clay on it, it doesn't really work to extract DNA from it. Another thing we found that we compared all our genetic data from the feathers to the blood samples, and we had very similar results, so that was assuring that we're finding something that's real, a representation of the population. As for like randomly sampling clay licks, it actually tells us the story about the population and not just of that little clay lick we're sampling. And as I mentioned, some of the very bad quality feathers, we couldn't use genetic markers for sexing, the traditional genetic markers. So we had to develop new sexing markers, and with these new markers, we were able to identify the sexes of even the degraded feathers up to like 80% of all the samples, which was quite good. And again, if you just find feathers and you don't actually catch the bird at the clay lick, you can be really sure which species it's coming from. So it's red and green macaw, the scarlet macaw, as you saw on the graph, they both had like red feathers, and if you just have two red feathers, you can't really tell them apart in the field. So you need some kind of genetic technique as well to be sure which species is that. So we used a sign-in test that really resulted quite good. You can see all the red and green macaws in one group, the scarlet macaw samples in another group. So that helped us to correct some errors that we thought like this feather belonged to scarlet macaw that actually didn't, and the genetic told that that's a different species. And also we used structure for this kind of analysis to separate the two species. So after the validation of the technique, we started with 30 genetic markers to validate on blood, and we find that only 5 or 6 genetic markers were necessary for the unique identification, which is quite remarkable because 5 or 6 genetic markers is really low, and it's quite cheap as well in the lab to use just 5 or 6 genetic markers. And we see like after like 5 or 6 you reach the plateau, you can recover more unique genotypes than the total number of the genotypes anyway. And we had family group samples as well, like from one nest where we captured the male and the female, so the parents, and then we also had the samples from the chicks. So it helped us to actually see whether we can distinguish the 2 different or 3 different chicks from each other and from the parents as well. So they're really powerful genetic markers. Altogether, we genotyped 137 scarlet macaw samples, so those are unique individuals, and 282 red and green macaws. And from all our samples, we find recaptures, so the matching genotypes in our samples, 13 for scarlet macaws and 23 recaptures for a red and green macaw. So what you can do with these recaptures, it's very interesting, because then you really want to know where these recaptures occur. So you find one genotype and you see the other genotype is matching in your Excel table, but then you want to go and rush and see on the map where those samples were coming from. And we had the maximum time, the time between the recapture was more than a year, so that was like assuring that these birds are coming back to the region or like living in the region, and we're recovering them for the next year and repeatedly in the next season as well. The maximum distance was about 30 kilometers, that the maximum distance between the recaptures, and it's about like this, so it's not really a huge distance. So that indicates something like they probably prefer to use one clay lick or one clay lick structure than like going every different kind of clay lick. Like if you think you just go back to the same supermarket to buy, and you're not like trying all the supermarkets just because they are run. So that's probably an explanation for the short distances for the recaptures. And using these numbers in capture mark recapture statistical analysis, you can also estimate the population size, where we just call here group estimates, because they're actually you're estimating the population size of the group size of that clay lick community. And we had quite reliable numbers between 25 to 300, so in the low hundreds numbers, the macaws visiting the clay lick, depending of course also the feature of whether it's a large clay lick, smaller clay lick as well. And if we combine these data, our group estimate sizes to density estimates that been done in the same region by just census. So we have a density estimate for the same species. We could calculate the radius that the macaws were coming from to visit that particular clay lick. So for example, here we calculated for this clay lick structure that they were coming about like in a radius of 4 kilometer distance. and we call that the catchment area of that clay lake. And this also like correlates with our finding that the recaptures are recurring just like within like 30 kilometers and no longer distances. But this is probably more like a local movement thing and not really affecting or segregating populations from each other. So it doesn't mean that they won't go and breed somewhere else with other macaws in the region, because we didn't find like regional genetic differences in the whole study site. And the end of the talk, I just wanted to come back to the Candamo that I mentioned at the beginning. So that's an intermontane valley isolated by the foothills of the Andes, about like 1,000-meter high mountains. It's a biodiversity hotspot. It's part of the Babajesonana National Park in southeastern Peru, which is like large, more than 1 million hectares protected area. And it also had a massive hydrocarbon deposit, the oil they find in the valley, and because of that, the government was thinking to cutting out of that region, out of the national park, like with 200,000 hectares, so they can sell the land for like oil lease. So that turned attention and conservation attention onto that valley to actually like look at why is it really, how important it is for conservation, but not really scientific research has been done in that valley before. So as far as I know, our genetic research was the first extensive research looking at on that valley as well. So for the first season, we went in the valley and also sampled macaw nest and clay lakes and get lots of samples from there and looked at we wanted to see the genetic distances between these and other samples in the lowland rainforest of Tambopata, and we actually find quite low but significant differentiations of the genetic makeup of these birds against those living outside of the valley. So we wanted to know why is it different? Why they different from the other populations? Because these were definitely not different from each other, but there was a sign that these are getting somehow isolated. And we looked at 3 landscape models, like it's called landscape genetic, when you try to use different landscape maps to explain the genetic structure of your population. So just overlaying different maps and then asking the question statistically which explains better the genetic structure. And the only model that came out to be significant was the elevation model that you can see on the map here. So it was really probably the elevation effect segregating these populations from the others. And we wouldn't have thought of that before, that high mountains could like stop a huge macaw that can go hundreds of kilometers from one place to another. But seems like they act some kind of restriction for the gene flow for these birds. And that might not be because of the high elevation, like they probably could easily fly it over, but it might be something like the habitat preference, that they prefer to live in a lowland rainforest, like which is inside the valley or here as well, but they don't prefer to travel to those high distance. They might get altitude sickness or something. So the outputs of the thesis, as I mentioned, we published 4 papers out of it, and the 3 last genetic chapters are written up as manuscript, ready for submission soon. We produced a documentary of the research, a 26-minute standard documentary that we collected with crowdfunding support, and we filmed 2 seasons of the research and made it into a documentary, because we find it's really important science communication as well to the outside world in not so scientific language. And we find that documentary is probably the best way to tell the story to a general audience. And the Hungarian version that's already done, and it's screening in Hungary now in the TV. I'm quite proud of that. And the English and the Spanish international version is coming early next year, so it's coming soon. So I did, of course, all these international conferences and different talks as well, like for local communities in the region about the importance of conservation in their land. And finally, the take-home messages of what we find in this study is that parrots are really a highly threatened taxonomic group that deserves some focused conservation management. In some places, we find, especially in the nootropics, it's important to preserve that high diversity of species there, and the most problem occurring there too. Artificial nests can be one of these genetic management to provide useful conservation tools for their survival. Not as much in Tambopata, because there you saw the huge land covered and continuously. They have lots of natural nests to select from. But other parts, like in Central America, they have lots of deforestation there. They already lost lots of trees and they don't have available trees. And macaws are actually critically endangered, for example, in Mexico. And these kind of same designs of nests that we already proved that they're using can be applied other parts like that for their conservation. And the genetic-based technique can reveal very novel information that otherwise unseen if you just go out to the field and collect data in a normal way. So with using genetic technique, we can have all kind of questions answered, and especially the non-invasive technique, like you don't need to capture the bird to get the genetic samples, you just go out and collect the feather samples, and already using that information you can have lots of data to analyze. And for example, one example is just to estimate the population size, which is quite useful for a field study by not even capturing the birds. So I would like to start my acknowledgements with my three supervisors. They're really great. Don Brightsmith, who is hopefully still on the Skype there watching us, helped a lot for the whole study setup and provided the home of the research and provided all the extra data we could also analyze to start with. And he also accompanied me to some of the crazy adventures that we went for the Amazon, sometimes with broken engine, etc. So thanks very much, Don. Rod Pickle took me over once I escaped from the Peruvian Amazon after 3-4 years, and supervised me in the lab and introduced me to all the wonders of genetics, and I really appreciate it. Just showed me like so much behind that I could just understand from the field. It was so much extra information I actually discovered here in the lab about my study species, and he eventually told me how to tell a great story after having all these months of heaps of information and genetic data to analyze. So thanks very much, Rod. And of course, this PhD couldn't have been done without my main supervisor, Rob Heinsohn, who was there for me all the time from the beginning, and also came down to the field and broke my engine.
Had a very uncomfortable night out on the riverbank too.
Yeah, that was his first night in South America, and spent his first night in my boat on the riverbank. So it was a great start. Thanks very much for all the academic help as well, and reviewing all the manuscript on the last night as well to put all these PhD together. And of course, I would like to thank all the other institutions that helped, especially Rainforest Expedition, who helped us all the logistic in the field to carry out, and all the other institutions that financially supported this research. And thank you very much for your attention. And if you have any questions.